Hello and welcome back to the Worm Podcast. I am so hyped for this one. If you are tuning in, I know you. You are likely already deep in the weeds, or should I say, deep in the Petri dish with C. elegans. You know the genetics, you know the cell lineages, you love the little guys. But today we are pivoting a little.
We are. Usually we're all about the biology, the neurons, the lifespan, all the genes. But today is about the hardware, the actual physical tools that let us even see that biology in the first place.
Right, because I feel like we take the tools for granted sometimes. We focus so much on the experiment, we forget about the equipment. But today we are doing a deep dive into a piece of tech that claims to solve what is, I think, arguably the single most annoying problem in worm research.
Oh, it's the problem that keeps PhD students in the lab at 3 in the morning.
And synchronization.
Synchronization.
It sounds so basic, doesn't it? Get all the worms to be the same age. But looking through the source material for this, it turns out that's incredibly hard to do without just ruining your data.
It's the fundamental hurdle. I mean, think about it this way. Imagine you want to study human development, right?
Okay, yeah.
Specifically, let's say, a hormone spike that happens during puberty. And you want to measure the average level of that hormone in a group of teenagers.
Okay, with you so far.
But your sample group isn't just teenagers. It's a room full of toddlers, teenagers, adults, and I don't know, grandparents, all mixed together. If you take a blood sample from that whole room and average it out, what do you get?
You get garbage, noise. The grandpa data is canceling out the toddler data.
Exactly. To get a clean signal, you need everyone in that room to be exactly the same age down to the month. Now translate that to worms. They develop so fast. A difference of just 3 or 4 hours means you're looking at a totally different biological state.
So if your synchronization is sloppy, your science is sloppy.
Precisely. And for decades, the field has been kind of stuck between a rock and a hard place on how to do this.
Okay, well, let's talk about those classic methods first, because I was reading the notes on this, and I honestly thought I was misreading the first one. It's called bleaching.
Hmm.
And I just assumed that was, lab slang for some complex chemical wash. But you're telling me it is literally bleach.
Sodium hypochlorite. It's essentially the same stuff you'd put in your laundry, just a lot more concentrated.
And what does that actually do to the worm?
Well, it's brutal. You take a gravid mother, a worm full of eggs, and you douse her in this solution. The bleach attacks the cuticle, the skin. It completely dissolves the adult worm's body.
It dissolves the mother.
Completely.
Yeah.
Gone. But, and here's the biological trick, the eggshell of a nematode is incredibly tough. It has this chitin layer that's like armor. So the mother dissolves, but the eggs inside survive.
That is medieval. I mean, it's effective, I guess, but it sounds like a trauma factory.
It is, and that's the problem. You get your eggs, sure. But then, to make sure they all start growing at the same time, you let them hatch in a buffer with no food. We call it starvation synchronization.
So, just so I have this straight, to get a clean baseline for an experiment, step 1 is dissolve the mother in acid, and step 2 is starve the children.
When you put it that way, it does sound horrific. But for decades, that was the gold standard. The issue is, if you're studying stress pathways or metabolism or aging, you've just started your experiment by traumatizing your subject.
You're poisoning the well before you even start drinking from it.
Exactly. Your baseline isn't zero. Your baseline is recovering from a chemical attack.
Okay, so that's the harsh way. I saw there was a second method, sedimentation. Sounds a lot nicer.
It is much gentler. It's just gravity, really.
Yeah.
Adult worms are bigger and heavier. The babies are lighter. You put them in a tube of liquid and just let them settle. The adults sink to the bottom faster.
So you just skim the babies off the top?
In theory. But in practice, it's messy. Physics is never that clean. Some adults float longer, some babies get dragged down. You end up with a mixed population. It's gentle, sure, but it's not precise at all.
So you have a choice. You can have perfect timing, but massive biological stress, which is bleaching. Or you can have happy worms, but messy timing, which is sedimentation.
And that is the exact dilemma that the technology we're looking at today solves. We're unpacking the Nemasync system. And what I love about this is that the solution wasn't biological. They didn't invent a new chemical. They solved it with mechanical engineering.
It's the Goldilocks solution. Precise but gentle.
Exactly.
So if I have this Nemasync set up on my lab bench, what am I looking at? Is it a robot? Does it have lasers?
No, and that's the beauty of it. It's a completely passive system, no electricity, no motors. It's just a 2-stage filtration unit.
Okay, walk me through it. I've got my plate of mixed-age worms, grandmas, moms, babies. I want just the babies, and I want them all born at exactly the same time. What do I do?
Step 1 is the stabilization filter. You wash your whole mixed population onto this mesh. Think of it like a sieve. The mesh size is designed so that the adult mothers, the gravid ones, get caught on top.
Okay, so the moms are stuck on the roof.
Right. But the debris, the bacteria, the old food, that all washes right through. So now you have clean adult mothers just sitting on a filter.
Step one complete. Clean moms.
Then step 2 is the harvest.
Yeah.
You take that filter with the moms on it and move it to a new dish that has buffer in it. The moms are submerged, they're comfortable, and because they aren't being bleached, they're happy.
And happy moms lay eggs.
Correct. They lay eggs right there on the filter. Those eggs hatch into L1 larvae. Now remember, L1s are tiny. The filter is designed so the L1s can just swim through the mesh.
Ah, so the moms are too big to fit through the gate, but the babies can slip right through the bars.
Exactly. They swim down into the collection dish below. And this is where the synchronization part really happens. You only leave them there for a specific window of time, let's say 15 minutes to an hour.
Okay, I think I see where this is going. If I come back in, say, 30 minutes and I take that bottom dish away, every single worm in that dish must have been born within that 30-minute window.
You've got it. You have a population that is defined by that time window. And because they hatch naturally and they swam through on their own, there's basically zero stress.
It sounds incredibly simple. I mean, honestly, it almost sounds too simple. Why is this a breakthrough? Can't I just go to the store, buy a nylon mesh or something, a coffee filter, and do the same thing?
I love that you asked that, because that is exactly what most people think. It's just a filter. But this is where we have to get nerdy about physics.
I am ready. Let's get nerdy. Unpack the filter.
So the creators of NemaSync spent something like 4 years figuring out why just buying a mesh doesn't work. And it all comes down to the microscopic architecture of the hole itself.
Okay, I want to push back on this a little bit, though. You're making a huge deal out of the shape of a hole. The notes talk about slits versus circles. Does it really matter that much? If the hole is small enough to trap an adult, it traps the adult. Why do we need semiconductor technology to make a sieve?
It's a fair question. It sounds like over-engineering, I get it. But you have to look at the math of the surface area and, more importantly, the manufacturing process. Let's look at a standard plastic filter, the kind you'd buy for a lab funnel.
Okay.
Those are usually made by a process called track etching. To make the holes, they bombard a sheet of plastic with particles. It's a random process. It's like shooting a shotgun at a wall to make ventilation holes.
So the holes are just scattered randomly across the plastic sheet?
Yes. And this creates what's called the doublet problem.
I saw this term in the source material. A doublet.
It's the killer. If you're punching millions of holes randomly, statistics dictate that occasionally 2 of those holes are going to overlap.
So instead of 2 small separate circles, you get 1 big figure 8.
Exactly. Or kind of peanut shape, that's a doublet. Or sometimes even 3 overlap, a triplet. Now, if you're filtering, I don't know, water, who cares? But if you're filtering worms, that doublet is a fatal flaw.
Because the adult worm, which is supposed to stay upstairs, finds that 1 double-wide hole and slips right through.
Precisely. And in a filter that has 1 million pores, you are mathematically guaranteed to have thousands of these doublets. You cannot avoid it with track etching.
So even if the manufacturer says these holes are 20 microns, that's just an average. The reality is you have a bunch of 40-micron failures just built in.
Yes. And if just 1 or 2 of those gravid mothers falls through a doublet into your baby collection dish, your synchronization is ruined. You've got contamination.
Okay, so plastic is out because of the randomness. How does NemaSync fix the doublet problem?
They stopped using plastic, and they stopped using random etching. They make these filters using semiconductor technology lithography, the same way Intel makes computer chips.
So they're building it like atom by atom.
Effectively, yeah. They use a high-purity nickel alloy, and they design the pattern on a computer. Every single opening is placed at a specific coordinate. There is 0 randomness.
Which means 0 doublets.
Absolute 0 defects.
Yeah.
Every single pore is identical to the one next to it.
Okay, I buy the no doublet argument. That makes total sense. But what about the shape? Why change it from a round hole to a slit? A long, thin rectangle.
This gets into the physics of flow and the material itself. Plastic is flexible. So if you push liquid through a plastic filter with round holes, the plastic actually stretches.
Ah, so a round hole might stretch into an oval under pressure.
Exactly. And suddenly, mom can squeeze through. Nickel is rigid. It doesn't stretch. But the slit shape is also about traffic control.
Traffic control.
Well, think about a round hole. It's pretty easy to clog, right? If one piece of debris lands perfectly on it, it's 100% blocked. But a slit, It's much, much harder to block the entire length of a long slit.
Right.
And for the larvae, the L1s, they move by undulating. They wiggle. A slit actually lets them pass through much more easily than a tight circle. It reduces the resistance, so you get faster harvesting with less clogging.
So the shape of the hole, just changing it from a circle to a slit, it actually changes the fluid dynamics and the behavior of the worm passing through it?
It does. It's a complete redesign of how a filter interacts with a living organism.
That is some serious engineering for what's basically a worm sieve, but I guess the proof is in the pudding. Or the Petri dish. Do we have data that proves this actually makes for better science?
We do. We've got a report from Nagy Bioscience that did a head-to-head comparison. Nemosync versus bleaching versus sedimentation.
Let's talk about the fertility data first, because we talked about how bleaching dissolves the mother. Does that stress actually carry over to the babies?
It seems to, yeah. The data show that worms that were synchronized with bleaching had significantly lower fertility when they grew up compared to the worms from sedimentation or nemosync.
So the trauma of being born via an acid bath has a lifelong cost.
It appears so. The nemosync worms had fertility rates that were nearly identical to the gentle sedimentation method, basically 100% healthy.
But sedimentation is the messy one, right?
Right.
The one with the bad timing.
Right, and that's where the nemosync really, really shines. They measure the standard deviation of age.
For those of us who haven't taken stats in a while, what does that mean?
It's the spread. If I say these worms are 90 hours old, how close are they actually to 90 hours? With bleaching and sedimentation, the standard deviation was around 0.12. With nemosync, it was 0.05.
So it's more than twice as precise.
Yes. That tightness means when you look at a population of worms, you aren't seeing a mix of developmental stages. You're seeing a true snapshot of a very specific moment in time.
And that precision is what lets you see things you might otherwise miss, which I think is the perfect segue to the paper we wanted to talk about, because this isn't just about making graphs look pretty. It's about discovering biological truth.
Right. This is the paper by Rasmussen and Reiner, published in the Journal of Cell Science. They were looking at a gene called MPK1.
Okay, so let's get to the payoff. They used this fancy nickel filter for this MPK1 study. What did they see that everyone else had missed?
They were looking at the vulval precursor cells. These are the cells that are responsible for building the reproductive organs. And the textbook definition, the dogma for years, was that MPK1 activation happens in one specific cell, the primary cell.
One cell turns on, everybody else stays off. Simple.
That's what we thought. Because when you looked at worms that had been bleached or starved, that's all you could see. But those worms were stressed out. Their signaling pathways were all noisy.
It's like trying to hear a whisper in a really crowded bar.
That is a perfect analogy. The bleaching created the background noise of the bar. The nemosync cleared the room. It made it silent. And in that silence, what they saw was that MPK1 wasn't just in the primary cell. It was in all 6 precursor cells.
Wait, all 6? So the textbook was wrong?
Incomplete. It wasn't just on in one cell. It was a gradient, a wave. The signal was strongest in the middle, and it just faded out towards the edges.
That changes the whole model. It's not a switch, it's a dial.
Precisely. It's a subtle, beautiful analog signal, but it's fleeting. It happens so fast. If your worms are even 2 hours apart in age, which happens all the time with sedimentation, you smear the data.
Yeah.
You completely miss the wave.
So you needed 2 things. You needed the worms to be stress-free to remove the background noise, and you needed them to be exactly the same age to catch the wave at just the right moment.
And that is why the tool matters so much. This wasn't a biological failure of the previous scientists. It was a failure of the plastic they were using.
That is fascinating. It really drives home the point that noise in an experiment isn't just annoying, it literally hides reality.
Exactly.
So let's wrap this up. We've gone from the chaos of dissolving mothers in bleach to the precision of nickel slits. What's the big takeaway for you?
For me, it's about questioning the standard tools we use. Nemosync realized that biology needed, engineering standards. By changing the material to nickel and the shape to slits, they solved a fluid dynamics problem that then allowed the biology to shine through.
It makes you wonder what else we're missing.
What do you mean?
I mean, we accept so many standard tools in the lab. Plastic petri dishes, rounded pipette tips, standard incubators. If simply changing a round hole to a slit can reveal a hidden gradient in cell development, what other discoveries are we missing just because we're using tools that were designed 50 years ago?
That is the big question. We tend to blame the biology when an experiment fails. But maybe we should be blaming the design of the plastic.
Something to mull over while you're picking your worms today. Thank you so much for joining us on this deep dive into the nuts and bolts, or should I say, the slits and nickel of worm science.
Always a pleasure.
If you enjoyed this look under the hood of the lab, please make sure to like, follow, and subscribe to the deep dive. It really helps us out. We'll see you next time.
Keep those worms happy.